Today I would like to present this manuscript that was submitted to bioRxiv last month, which is of interest for our work here at Liber, and especially because they actually used as a main data was our LIBD DLPFC RNAseq phase 1 samples. So the paper, this is about isoswitching related to aging in human brain tissue, examined by as explored by this paper. Now, just to review what is isoswitching, they use this as a shortcut for what is more commonly known as isoform switching, and there are a lot of literature. There's a lot of literature about this, and it's related to a statistical significant change of the isoform fractions that can be observed in a gene. As the expression of a gene is the sum of all its isoforms expressions, the isoform fraction, or IF, we'll see it in some of the diagrams here, is a relative expression of an isoform in the gene. It's simply the proportion of its expression from the total gene expression. And this illustration actually comes from a very interesting tool that's been around for a while. It's called the bioconductor package called Isoform Switch Analyzer by Christopher Whitting-Syrup. And it can take output transcript information from various tools, including string-tie and many other transcript quantification tools to analyze this isoform switches using various methods. And as an example here, to understand that it's important to see the importance of what is called differential transcript usage analysis, which is shown here. If you look at these three bar charts below, when we have like these three isoforms, you can tell that just gene expression might find non-significant expression levels, non-statistically significant. If you go to isoform expression level, which we know, we can see that there's significant expression differences, but even we can find even more subtle signal if you look into each gene separately in terms of isoform fractions changes from one condition to the other. So it increases significance in some situations. So the isoform switching events are what this paper looks into, and specifically in brain tissue. Now, the other topic, that the main use of these isoform fractions and isoform switching in this paper is to propose an age prediction model. So, or model how expression change related to age are reflected in a transcriptomic landscape. And there are actually many age prediction models from RNA-seq expression in the literature, and only a few of them are shown here. Most of them are based on gene level expression, as we expect. So with some exceptions were listed here, like there was one of these papers, the first one. It looks at actually splicing events, but they don't disentangle these exon-exon splice junctions into actual consistent transcript structures. They just looked at the junctions there to try to associate with age. And this is usually referred to in the literature as a transcriptomic age clock. So it's a way to build a model based on the expression changes with age, and be able to even predict looking at a sample. That's the ultimate goal, right? We look at a sample, RNA-seq sample, and predict the age of the individual sequenced in that sample. Now, Beryl print here takes a fresh look at this transcriptomic aging clock models by analyzing directly the isoforms, and specifically the isoform fraction changes. So it uses transcript level quantification first, and they used, as I mentioned, LIBD DLPFC RNAseq phase 1 samples, and in those samples they quantified the extended set of transcripts found in the latest version of the CHES3 database that the Center for Computational Biology at Hopkins, they also built. And a quick refresher, which is CHES3 is based on a combination of reference transcript databases which were curated, and the set of transcripts included there was augmented with evidence-based isoforms, novel isoforms obtained through assembly with the string tie. of read alignments from almost 10,000 GTEx samples. So these transcripts were quantified with Salmon, and then the first step was they split it into prenatal and postnatal samples to perform a DTU testing with their program called SPIT here. Splitting iteratively, it's probably a way to look at the methodology they use for testing. And sorry, this was a simple DTU analysis, differential transcript usage analysis between prenatal and postnatal at this first stage. So it was treated like a case control DTU analysis. So again, this is just the initial stage. It doesn't do any age prediction per se at this level. It's just a simple postnatal versus prenatal. You could do a prediction based on that, but basically they observed a sort of systematic isoform shifts around birth, as seen in the PCA here, some of the figures and other figures that present at the beginning for this part, but that they show a clear prenatal postnatal separation. And these significant DTU events are observed for over 5,000 genes they found, and they use this SNAP25 gene as a prominent example here when the switch from 25A to isoform switch from SNAP25A to SNAP25B is highlighted in this section of the preprint. Now, going back to the age prediction model, this involves, of course, all the samples from these DLPFC samples across an age range from prenatal to 85 years old. So initially they started with a random exploration using random forest regressor model. And this is relatively simple learning framework where they use the 100 trees to avoid overfitting. It showed that the age can be predicted with over 80%, 85% accuracy just by using this direct random forest approach. Now, as you know, random forest is an ensemble learning method composed of multiple decision trees where the final model output is typically an average of or aggregation of the predictions from these individual trees. Next, they consider, so the idea was that, yeah, they could do better, and they knew that, so they tried to refine this model. And first they tested the robustness of the method, and they use this feature ablation technique where you can remove the most important genes and isoform fractions observed there and retrain the model. And that showed that even after removing up to 5,000 genes, I think, they can still get some signal about some relatively good association with age, which actually was a way to show that the model is robust and isoswitching is widespread in brain tissue, and it's not concentrated in just several genes that would drive the model. They also, another refinement was to use the log transformation of the chronological age, which of course this transformation gives a greater weight to early expression changes where we know they're actually very large in the early years. And this improved accuracy. And by feature ablation was also a way to look at the feature importances, and it was indirectly a way to refine the feature selection. And in the end, they selected 100 genes, top 100 genes in terms of feature importance, which included 341 transcripts. And this has achieved a high accuracy of R-square 0.99798 almost. Yeah. So they chose these 100 representative genes and 341 transcripts from now on to further refine the model. And that's an interesting development here. Besides refining the random forest regressor as described using log transformed age and this selected features, the output, the initial prediction from this one was further sent for refinement to this lasso regressor stack, which is an interesting contribution to this methodology. So this is a layered approach, right? The first layer is the random forest regressor trained on the log transform ages using isoform fractions from the top 100 genes. And the second layer is takes this age prediction and sends it to this one or more of the 5 overlapping lasso regression models. This is a multi-stage framework which seems to have allowed for the identification of DTU genes with a strong predictive power within specific age intervals. By using overlapping age ranges, as we notice here, the ranges are very much overlapping. The justification here is what all this we can better reflect the age continuum and bring overlaps between adjacent stages, life stages, and capturing better the smooth transitions instead of treating developmental phases as discrete stages of life, of age expression changes. And many samples fall within the overlapping range of the two lasso regressors, so allowing for interpolation from both models, and they take the average in that case when two predictions are given. Now, one of the figures on the paper, they explain this example and showing that, as for example, an adult sample represented here by the red triangle, the base here, is traced through the model pipeline. And first, each tree in the random forest predicts the sample age, and these predictions are averaged to produce the final estimate. And based on this prediction, the sample is then routed to one or more of the 5 lasso regressors, each corresponding to a specific age range. In this example, the prediction falls within the range of both L3 and L4, so overlaps these two ranges here, bottom, receiving predictions from both. And as I mentioned, the average value is used as the final prediction. Now, of course, the question would be why, and it's interesting idea that they already had a good prediction from the refined random forest model. So why would now go through this lasso and lasso regressors models? And there are some good justifications providing in the paper that the idea is that different from those 100 genes, since lasso regressors are linear models, they are easier to interpret, and also different genes have different importances or coefficients at various stages of life. And so different genes matter differently at different life stages. This was a way to allow for building different linear models and some overlapping and having smoother transition between various stages. Now, there was some validation on independent data. One good validation was on similar random forest model trained on repeated training, and the whole approach was produced on a data set of 53 human forebrain samples, and they got this R-square of 0.992. But interestingly enough, they tried to reproduce the model in heart samples, left ventricle heart tissue samples, and the performance here for this model was very, very poor, unexpectedly low, just 0.08. And I think the idea here is, there are multiple possible explanation here that heart tissue might have a very different aging model and very different from the brain tissue. And one kind of obvious possibility there would be that there are not so many isoform switches, but there could be like just the differential, just gene expression switches. And to remind you, the model he used here for is DTU, which doesn't capture difference between transcripts across genes. It only looks at how isoform fractions change within each gene separately. So this model does not capture basically DTE across genes, different transcript expression, which might explain why maybe heart tissue has a different aging model. Now, they also tried reproducing the method on the macaque transcriptome, but there was a difficulty here, of course, because in terms of transcript annotation and genome annotation for this species, so they actually kind of rebuilt their own annotation in order to get a better input for the model. And they had to focus on some of the strongest predictive switches here, which were found in these two genes like PALM and SNAP25. And they did observe a similar isoform switching patterns like in human, which suggested this evolutionary conservation of iso switches across age ranges. Now there are quite a few figures in the paper showing how the impact of this and the relevance of looking into isoform switching when it comes to functional implication, changes that they do in protein structures, and they show the figures. I forgot to include them here, but with protein structure predictions for some of the isoforms that showing domain loss, protein domain losses, and potential implication in this functionality of biomolecular changes that are observed due to isoform switching. So the idea being that they are not, isoform switching is not an abstract, just abstract changes in RNA, but they lead to concrete modifications in the resulting protein, and they can, by altering functional domains, they can impact biological pathways and cellular functions, and can help in providing a better understanding of the molecular underpinnings of brain development and aging. I guess that's it for now.